let's talk about the highly diverse yet sometimes overlooked world of eukaryotic microorganisms. I'm going to begin by drawing a eukaryotic cell. We're going to make this just a very general eukaryotic cell, knowing from your pre-party activity that there is a lot of biodiversity within the eukaryotic world. Maybe we can name our cell fill or something like that. And one wonders when eukaryotic cells sometimes lack an external cell wall, where they get their structure. So let's draw some in this one region of the cell, some of what gives a eukaryotic cell its structure, and look at what we call the cytoskeleton. The cytoskeleton is comprised of three major components. Now we'll go ahead and label these different components. So these large blue ones that I ended with drawing, these are microtubules, and microtubules are about 25 nanometers in diameter. These are the largest of the cytoskeletal components, and microtubules function in a lot of diverse ways. One of the things that a microtubule does is that it serves as sort of the highway within the cell. That is, these provide the network along which vesicles can travel as they're taking their cargo from one place to another. I like to think about the eukaryotic cell as being a bit like a cell city. These would then be the highways of the cell. Microtubules also play another important role. They come together to form the network that creates flagella within our eukaryotic cells. And so the core components of a eukaryotic flagellum are packed full of microtubules. If this is a flagellum extending from one end of the cell, then microtubules, in fact, will be the central components within that, and we'll learn later about the fact that there are many microtubules in a pattern called the 9+2 pattern that extend out through, and I won't be able to get all of them in there, that flagellum to allow it to have its motility and structure. So that, again, is something that microtubules do. Is they provide the structure and the framework to the cell, so they give the shape to the cell. And we know that shape fits function. And so if a cell loses its microtubules, often it wanders aimlessly because if in the absence of its form, it fails to provide its function. And it's important to note that microtubules are made out of a protein called tubulin. So this protein, actually, if we were to zoom in even further here, so I'm trying to show that this is a helical structure where the subunits alpha tubulin and beta tubulin wrap together to form a hollow cord helical structure that comprises the microtubules. Let's take a look and maybe zoom in a little bit at the next cytoskeletal component, the microfilaments. Microfilaments are very, very different, and I've tried to form some networks of microfilaments, hopefully showing these are very, very small, but they often form groups. And microfilaments tend to be around 5 times smaller, so between 4 and 7 nanometers. And they actually are comprised of actin. So actin, basically you've got this double cord of actin where there's one kind of component there and then the next that wraps around it. And so we'll try a little bit different color here. So this kind of wrapping cord of actin. Now these play a very important role in the amoebic kind of movement of a cell that does move via these false feet or pseudopodia. They also play a really important role in food acquisition through phagocytosis, or if you're talking about a eukaryotic cell like a white blood cell, then they're playing a huge role in acquiring foreign invaders and bringing them into the cell. In this awesome video taken by a friend of mine when she was teaching general biology, you can see that it is an amoeba, and this amoeba has pseudopodia, which also means false feet, but it's a way to say that they form these large surface projections with streaming cytoplasm that allow them to reach out and engulf some of the surroundings. Now, in this case, this amoeba is reaching out to try to acquire food, and you'll watch as the actin within the pseudopodia polymerize in order to enable it to reach out and try to grab the surroundings. This is awesome. I love seeing the streaming cytoplasm. We'll do it again. So it's reaching out, right, and trying to grab the nutrients in its environment. And this likewise could occur within a human system when a white blood cell would reach out and try to grab a foreign invader in order to degrade it and get rid of it. And what's really interesting, though, is that some foreign invaders, like the pathogen Listeria monocytogenes, it actually exploits actin polymerization. So it actually sends out a signal to cause actin polymerization, and it rides on the actin as it polymerizes. It's its means of getting around the cell. So it's like riding the actin as it would ride a roller coaster to get around quickly around the cell that it is invading. Our final cytoskeletal component is the intermediate filament, not surprisingly. Approximately 10 nanometers in diameter, and it actually is very hard to draw because it looks like a rope within a rope. So it's as if you've got these components that are like several ropey sort of networks. And then if we were to look at the ultrastructure of this, we would see that inside of this ropey-looking structure, there would be more ropey-looking structures. And these intermediate filaments are important in stability. They're very strong, and they're made of the protein keratin. So that's something you're pretty familiar with, right? Your hair and your fingernails, very strong and provides a lot of stability. Let's now move to the nucleus of the cell. The nucleus has both an inner and an outer membrane. Together, these comprise what is called the nuclear envelope. Now, spanning the nuclear envelope are nuclear pores. These are relatively large, actually, very large. They're like highways compared to the pores that we've seen before in the cytoplasmic membrane, making the nuclear envelope far less exclusive. It lets a lot more in and out. And in fact, this nuclear pore complex right here is comprised of proteins and the pore, and large things can pass in and out via these nuclear pores. Just subsurface of the nuclear envelope is a network of intermediate filaments. I'm not going to draw all of these intermediate filaments, but if you would like to, I highly suggest spending some artistic time. These obviously provide support. Another component of the nucleus is called the nucleolus. Now, the nucleolus is not membrane bound, but if you were to look in an electron microscope, you would see that it was a very recognizable region. Cells might have one or they might have many, but they're more visible when a cell is not dividing. And obviously throughout the entirety of the nucleus is our chromatin. We know that chromatin is technically defined as the network of DNA and proteins that form eventually, and during a division, the chromosomes. Now, we can define chromatin as being one of two types. We can either see that it is heterochromatin, or euchromatin. In a cell that is not dividing, we see euchromatin, which is fully extended out, and we'll define that specifically here in a moment. But heterochromatin is much more tightly packaged, very highly wound. We know people like that, right? And heterochromatin hides genes because they're so tightly packaged that it's very hard for the machinery that reads the genes to get to them. So they're inaccessible. And if you get real sexy minded, you can think about what that has to do with the field called epigenetics. So when we look close at chromatin and we zoom on in, we can see what it would look like, and I want to show you the base level structure, where if we were looking at euchromatin, this is what it would look like. It's often termed beads on a string, because if we have our DNA here, our DNA wraps, in fact, about 1 3/4 times around a protein complex. And in this protein complex that will make a different color, this protein complex is called a histone protein complex, what's almost like a spindle around which the DNA wraps. So to do better in my drawing, I might try to show that this DNA, okay, so it goes behind the histones there, and then it comes out and around and is external right there. So it's wrapping around this histone core. And we call this a histone core octamer. The reason we call it that is because it's comprised of 8 histones, 2 copies of 2 different types of histones. Now, there is also another histone. I sound like Star Wars, right? There's yet another histone. And this host histone is called H1. And H1 associates with the linker DNA. This whole region here that includes the linker DNA, the histone core octamer, the DNA wrapped around that is termed the nucleosome. And we think of the nucleosome as being the core packaging unit of eukaryotic DNA. Now, I've created a model. You might find that it's valuable to create a model too to help you better understand this. My garden hose bit the dust one day when I decided that it looked like it could be a histone core octamer. So this garden hose here is the histone core, called H2A, H2B, H3, and H4. And so you've got different components of that histone octamer. And then you've got the DNA base pairs wrapped around the histone core. There's 146 base pairs wrapped around the histone octamer and 54 base pairs of linker DNA. Now, linker DNA is associated with the H1 histone, and you can see that if an H1 histone were to interact with another H1 histone, that could lead to more packaging. So we might say that this beads on a string orientation of DNA is the least packaged that it can be, where the genes here are very accessible to expression, whereas as they package down further and further and further, and there's more and more condensation that can happen until finally at the highest condensation they're forming chromosomes, we recognize that that packaging, it takes what would be just vast amounts, remember that exercise we did for prokaryotic cells, vast amounts of DNA, and packages them down into something that is much more manageable, particularly in a dividing cell. Let's keep drawing. This is getting fun. We know that the nuclear envelope is actually part of what is a contiguous membranous network. And in fact, we see that the nuclear envelope is contiguous with the endoplasmic reticulum. And right here, I'm drawing one type of endoplasmic reticulum, and you'll probably guess that what I'm drawing is the rough endoplasmic reticulum, or RER, because I'm studying it with ribosomes, and you can see there all of the ribosomes that are on the surface of this rough endoplasmic reticulum, which gives away the fact that these ribosomes, which are 80S ribosomes comprised of a 60S and a 40S subunit, are synthesizing proteins. So what's happening is they're reading RNA and using that RNA to make proteins, and these proteins will fold inside of the rough endoplasmic reticulum, and they'll gain that secondary and tertiary structure inside. Now, these proteins are very different than a protein that would be synthesized by a ribosome out in the cytosol, because these are going to be either secreted outside of the cell, or they're going to be delivered to the membrane, and they're going to be membrane spanning proteins. So within the rough ER, they are made and they fold, and then they take a membranous route via vesicles that will bud off of the rough ER, and these vesicles will be carrying the protein. So now this protein will further have its journey through the remainder of this membrane network, where it will head off, again carrying its protein cargo, and it will merge with the Golgi apparatus. Now, the Golgi apparatus, we may see many of these organelles within a eukaryotic cell. If a eukaryotic cell has maybe a lot of proteinaceous scales on the outside, then we know that it's going to secrete a lot of proteins and will have an extensive network of Golgi apparatus. Or we may see that a cell that doesn't secrete many proteins at all lacks a certain amount of Golgi apparatus. It's really not a big component of what it does. But if a cell does export a lot of proteins, of course, we'll see extensive flat sheets of Golgi, where this protein, now destined to the Golgi, will merge with the Golgi. And the Golgi actually is an asymmetric organelle, meaning that it has two different distinct sides to it. So one of the sides is called the cis or forming side, and the other side is called the trans or maturing side. So if we picture that this protein now takes its journey through the Golgi, and within the flattened sheets of the Golgi called cisternae, this protein will become modified. So as it's taking its journey through this organelle, it might gain things like phosphorylation. A phosphoryl group might be added, or glycosylation, that is a carbohydrate moiety, might be added onto it. Now, and I kind of think about in our cell city analogy, that this is like the factory, the export factory, and it's going through these quality control and modification stages, and now it's ready to be sent in a vesicle with its modifications. And of course, we know it would be actually following a microtubule. That vesicle would be using a microtubule as a highway, and then it would eventually take its cargo. And let's just say that this particular protein is a membrane protein. So now it will insert itself into the membrane with its phosphoryl groups and its glycosyl groups, its carbohydrate regions. So it's a glycoprotein, it's a transmembrane or integral membrane glycoprotein, and it will do its job there. So these organelles all together, the nucleus, the rough endoplasmic reticulum, and the Golgi apparatus are part of what we call the endomembrane system. Now, additionally, we can add to this endomembrane system, and some of you might have been recognizing that there's another kind of endoplasmic reticulum. This type of endoplasmic reticulum looks a little more like a tubular network than it does like these cisternae flattened sacs. This is called the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum is the site of lipid synthesis. So like proteins, lipids that might be exported or utilized external to the cell may be synthesized in the smooth endoplasmic reticulum. Some fungal proteins also utilize the smooth endoplasmic reticulum for calcium synthesis and storage. So depending upon the type of cell, we may see some diversity in that function, but it's associated largely with that lipid synthesis. So this together is our endomembrane system, but I should also make note that a vesicle, some of the vesicles that are synthesized actually can become another organelle. This other organelle is called a lysosome, and a lysosome in our cell city, I like to think about a lysosome as being like the garbage compactor or recycling center, where it's really taking care of the waste. And so anytime a cell brings in a bacterial pathogen that it's trying to either eat or kill, for whatever reason, it will bring it to the lysosome. Inside of the lysosome is where it's degraded. So that makes you think, gosh, that lysosome sure must be packaged with a lot of enzymes that break down macromolecules. So I want you to make a hypothesis as to what those enzymes might be, remembering that we commonly would put the macromolecule, and then to make an enzyme name, we would add ACE to the end. So if we thought about breaking down a protein, what would we call that particular enzyme? And that lysosome is filled with those. It also has a very low pH, so that acidic pH helps to further the degradation or breakdown of the macromolecules. So thinking back to all of our macromolecules, what are all the ACEs that we would find in the lysosome? And we'll open our class by thinking about that question.